Hello, my name is Dr. Ortiz-Gonzalez. I am a pediatric neurologist at the Children's Hospital of Philadelphia, and I also run a neuroscience lab. Today, for this video abstract, I would like to share our work most recently published in Brain Communications titled Lysosomal Dysfunction Impairs Mitochondrial Quality Control and is associated with neurodegeneration in a rare disease called TBCK encephaloneuronopathy or TBCK syndrome. The first author of this work is Dr. Jose Tintos Hernandez, and I am the senior author. The first observation that we published in this paper is that there is upregulation of mitophagy in TBCK fibroblasts. Mitophagy is a process by which mitochondria that are senescent or dysfunctional are normally cleared in the cell, and the organelle that is responsible for ultimately degrading them is the lysosome. In this experiment, we can see lysosomes labeled with lysotracker red, mitochondria in green, and when we merge them, we see an image in healthy cells like here in this panel, and we appreciate that in TBCK fibroblasts, there is a robust upregulation of mitophagy, as evidenced by the high degree of colocalization. We quantified this in panel C here, and we also quantified the number of lysosome in the cell by using, again, lysotracker. So we have a particle count per cell, which is significantly increased, as well as the area of the lysosome, suggesting that the lysosomes could have a larger size. And this is confirmed by flow cytometry by looking at LAMP1 quantification. We then ask if the mitochondria in the cells are healthy, or is there any signs of mitochondrial dysfunction. And in this figure 2, we show that there is reduced mitochondrial DNA content, that there is a suggestion of decreased mitochondrial biogenesis, which is controlled by PGC1 alpha. This is an RT-PCR looking at mRNA levels for PGC1 alpha. In panel C, we look at respiration capacity at baseline, which is decreased, as well as upon FCCP stimulation by using seahorse assay. In panel D, we also show decreased maximal respiratory capacity. Furthermore, we started examining whether we could correlate the degree of the clinical severity of the neurological symptoms with any features in the cells, specifically with mitochondrial respiration or mitochondrial dysfunction. In panel A, we show that there is indeed decreased respiratory capacity, as we previously mentioned, but that there is a more marked phenotype in the severe patients versus the mild patients, shown in gray versus orange for severe. And I should say the severe patients are the ones that are homozygous for the 126 truncation mutation or the WOD1 mutation. It is interesting that when we look at maximal respiratory capacity, we can appreciate that there is a significant difference between control and mild patients, but furthermore, that if you compare mild versus severe, that there's also a significant difference in maximal respiratory capacity. We also observe a correlation in mitochondrial DNA content. So when you compare controls, again in blue, with patients that are mild by definition of known evidence of neurodegeneration versus the severe patients, there is no significant difference between control versus mild, but there is a significant difference between controls and severe TBCKE patients. Furthermore, we ask whether this difference in mitochondrial DNA content could be because there is differences in the amount of mitochondria per cell, and for this we labeled the cells with pouring or VDAC and did flow cytometry to quantify mitochondrial content. And although there is a trend, there is no statistically significant difference. We do see that if we look at reactive oxygen species quantified here by cell rock screen, we do see that there are least amount of ROS in controls, but significantly increase in mild and further increase in the severe patients. We then ask if the dysfunction in mitochondria could be because there is impairment in degradation, which is ultimately completed at the lysosome. So for these assays, we look at markers of proteolytic activity, and this is a DQBSA light fluorescent imaging showing decreased DQBSA activity in TBCK cells, which is quantified by flow cytometry here. And then we also look at cathepsin D, which is another of the enzymes present in the lysosome, which we do see significantly decrease in TBCK patients. Another marker of lysosomal dysfunction that we often see clinically is lipid accumulation. So we also evaluated for neutral lipid accumulation, visualized here in green by the body P493 dye. And again, the controls are on the top and the TBCK panel is in the bottom. And also looking at unesterified cholesterol accumulation, which is shown here in blue in control versus TBCK. This is the merge panel, and this is a quantification of the Philippine and the body weight by plate reader assays. Finally, we we ask if the mitochondrial dysfunction is due to intrinsic mitochondrial pathology, or could it be because of the impaired lysosomal degradation. And in this experiment, we attempted to answer that by treating the lysosomes with acidifying nanoparticles. This will reduce the pH in the lysosome and increase its degradation efficiency, regardless of what the mechanism of how TBCK may be affecting the lysosomes. So here we repeated the CHOR assays that we mentioned before, and I will bring your attention to, again, the control in blue. Orange, we have the TBCK cells, and if we treat the TBCK cells with acidifying nanoparticles shown in the black trace, it essentially reverts the respiration to control levels. This is quantified in the panel on the right, and the B panel on this figure is just to show you that the nanoparticles indeed colocalize in the lysosomes. So in conclusion, our work shows that mitophagy is upregulated in TBCK fibroblasts at baseline, that there is respiratory dysfunction, mitochondrial dysfunction in TBCK cells, and that seems to correlate with the severity of the clinical phenotype, meaning that patients that have a neurodegenerative course have more severe mitochondrial dysfunction. We do find evidence that the mitochondrial dysfunction may be secondary to lysosomal degradation, and that it could be improved by lysosomal acidification strategies, which is an important point to continue to further investigate to explore if this could have therapeutic value in the future. Thank you for your attention.